All right, so we've been talking about just the normal inheritance of different genetic traits, and remember that all of the genes that you have are stored in your DNA. So all the genes that you have are stored on the DNA that you have. Okay, and so far we've just been following normal patterns of inheritance where everything is going, according to plan. But as we all know in life, not everything always goes according to plan. Okay, so what I want to talk about today is when things can go wrong or just go differently with your DNA. Okay, so what we're talking about are changes that can happen in your DNA. And when we talk about that in biology, what we're really talking about are mutations. Okay, and all a mutation is, it's just a change in an organism's DNA. Okay, so it's just some kind of change in the nucleotide base sequence of an organism's DNA. And mutations can happen for any number of reasons. Sometimes they just happen naturally, or exposure to certain chemicals, or maybe some kind of radiation, UV radiation, or X-rays, or something like that can also cause changes in DNA. Okay, and I want to focus on two main types of mutations today. The first type that I want to talk about is what's called gene mutations. Okay, and so this refers to mutations that happen within an individual gene on a chromosome. Okay, so let's start off. We have, say, a chromosome. Okay, so let's say, we're just talking about one single chromosome that you have. Okay, and remember that a chromosome is just a structure inside your cells, located inside the nucleus, and it's made up of DNA wrapped around several different proteins. Okay, and so since this chromosome is made out of DNA, all your genes are located on the 46 or the 46 chromosomes that you have. So let's say on this one chromosome we have one gene here, and maybe this is, the gene for eye color. And then, somewhere else on the chromosome we have a different gene, and maybe this is, something like hair color. Okay? And so in a gene mutation, what you get is an actual change in the sequence that makes up a single gene. All right, so let's say we talk about the gene for eye color. All right, and let's say we examine it and we just look at a small segment of the DNA sequence that's in that eye color gene. Okay, so let's say we start in our DNA, and normally the DNA sequence in your gene, maybe it looks like, and I have this written out somewhere, but maybe let's just say the DNA sequence is TAC, AGT, and then CCA, and so on and so on. Obviously, the gene is going to be much longer than this, but we're just going to look at a small segment of it. Okay, and remember, these letters, they represent the nitrogen bases within the nucleotides that make up the DNA. This is the actual coding sequence. Okay, and remember, we talked about earlier that genes are responsible for making proteins, right? And proteins are responsible for building your traits. Okay, and the way that we make proteins using our DNA, the first thing we have to do, we have to use that DNA to make a strand of mRNA. Okay, so what would base pair with or what mRNA would be made from this DNA strand? We can find out by what base pairs. T gives us an A gives us a U, C gives us a G, so on and so on. I'll put in U, C, A, and then G, or that would be a G here, G, G, U. Right, and this would just be a short segment of our mRNA that's made using our DNA. And then finally from this, we can get the amino acid sequence out of here because DNA is used to make proteins, and amino acids are the building blocks of proteins. And so what we would use here is a little codon chart, and actually I have one on hand. So if I look it up, this particular DNA sequence produced the amino acids methionine, which I can just write as MET. The DNA AGT gives us mRNA UCA, and that UCA gives us serine, so SER, and then this GGU gives us glycine, GLY. So this would be like the normal DNA sequence that would happen, right? So let's say we can label this as a normal occurrence, all right? But in a gene mutation, we have some kind of change within this original DNA sequence, okay? And there's two different types of gene mutations that can happen. Okay, so the first type I want to talk about is a substitution. Okay, so a substitution mutation. And literally what that means is that one of these nucleotides is substituted with another one. Okay, so let's say we have just a substitution. So let's say we start off with our DNA sequence T, A, and then now where we originally had C, let's say we substitute that in with a G. Okay, so now we have TAG, and then the rest of the gene is unaffected. So we have TAG now, and then we still have AGT, and then CCA, and then so on and so on. Okay, so that's our DNA molecule, and so the mRNA that would be made using that now is also going to be changed, right? Because the mRNA is made using the DNA, and so now instead of AUG originally, now since we have TAG in the DNA, it would give us AUC, right? And then everything else would still be the same because everything else was unaffected. So UCA and then GGU, right? So in this substitution mutation, where we originally had this sequence here, right? We had a C originally in the DNA that gave us a G in the mRNA that gave us this amino acid MET. Now we have this G change in the DNA, which causes a change in the mRNA made. And then let's just see what happens in the amino acid sequence that we get. And so I'm just going to abbreviate amino acid as AA. And if I look it up in the chart, now instead of methionine being produced, we get isoleucine, right? So ISO, and then everything else stays the same because it hasn't been changed. Gly, and then so on and so on. So you can see in a substitution mutation, wherever it gets substituted, that one amino acid is going to be changed, okay? And so the protein that's made is going to be different because the amino acid sequence is different. So now it might be a different trait, you know? Instead of being the protein that gives you blue eyes, maybe now it's the protein that gives you brown eyes. But in a substitution mutation, only a single amino acid or a single DNA, or I guess I should say a single nucleotide is replaced by another one that affects the mRNA that's made, and that'll affect the amino acid that gets made. Okay, so that's one type of gene mutation. It's a substitution. And it's bad, but it's not as bad as the other type that I'm going to talk about next. So the other type I want to talk about is an insertion or deletion. Okay, and what happens here is that instead of just switching out a DNA nucleotide, now you actually insert or delete one of those DNA nucleotides. And we'll see why that can be particularly bad. Okay, so let's start with our DNA again. All right, and then remember originally we had our DNA sequence TAC, AGT, CCA. Okay, and let's say we want to insert a nucleotide right here, right before this first, or this A right here. Okay, so now our new DNA sequence, if we insert that new nucleotide, we'll still start with TAC, and now we have an extra A, right? And then we still have our original AG, and then TCC, and then A. And remember, I'm writing it out 3 at a time because that's the way that our cells read this DNA molecule, right? Or I guess to say they read the mRNA molecule that way, but they read it 3 nucleotides at a time, and that's called a codon, and those 3 nucleotides at a time give you a different amino acid. Okay, so now I've inserted this A within the middle of our original DNA sequence, so let's see what kind of change that'll have. Okay, so again, we still use our DNA to make a molecule of mRNA. Okay, so now we still make AUG, and now with this extra A, we make a UUC, and then now we make AGG, and then this A now gives us a U, and then it'll continue on and on. We're just looking at a little piece. But now let's see what happens to the amino acids that are made. All right, so now AUG will still give us MET, which we started with. Now UUC gives us something different than what we had. UUC will actually give us phenylalanine, a different amino acid, Phe. So this amino acid is affected, and then the next one will also be affected because what it does, it throws off our reading frame. Since we're reading it 3 at a time, it changes all of the rest of the mRNA sequence, so we're reading everything differently. So now this AGG gives us arginine, which is ARG. Right, so in an insertion deletion, now not only is the place of insertion, the amino acid there affected, all of the following amino acids will be affected as well. So the second and third amino acid and fourth, fifth, maybe up to even the 100th amino acid, they'll all be affected from what we originally had. Whereas in a substitution mutation, only the place where it's substituted gets affected. Okay, so you can see that insertion mutations are much worse than substitution type mutations alone. Okay, and then you can think of the same thing happening with a deletion mutation, right? Let's say in a deletion, we just delete, let's say the C DNA nucleotide, and so this A will shift over here, and then the whole reading frame will be shifted as well for a deletion. So all of the subsequent or following amino acids will be affected for a deletion as well. So substitution mutations, they're bad, but they're not as bad as an insertion or deletion mutation, which can affect all of the amino acids or a lot more of the amino acids that are coded for by a particular gene, which can really affect the protein that's made, which will really affect the trait that's built. Okay, so these are all examples of gene mutations. So the three types of gene mutations, right? So gene mutations, we can have substitutions, insertions, or deletions. Okay, and these mutations, they can happen in any of your cells, right? They can happen in your skin cells or in your bone cells or in your muscle cells, and they can also happen in your sex cells, and that's particularly bad because your sex cells, your sperm or your egg, these are the sex cells or the gametes. These are used to pass on your DNA to the next generation, and so if you have a mutation in the DNA in the sperm or an egg, that'll affect all of the DNA that's in the new baby that's formed, in the new zygote that's formed when the sperm and the egg unite. Okay, so now instead of just a mutation that would happen in your skin and would end there, if you have a mutation that happens in the sperm or egg, now the new baby or zygote that develops from that sperm or egg will have that same mutation in all of its cells. Okay, so mutations are only passed on. Mutations are passed on when they occur in sex cells. Okay, and that's a really important point, right? That's why if you go to the dentist or the doctor and they give you an X-ray for whatever reason, they put that lead apron over your sex organs, so to protect the sperm and the eggs that you have or are making. Okay, that way you won't pass on any mutations that could be caused by that X-ray or whatever procedure. It prevents mutations happening there, so that you don't pass on those mutations to your offspring. Okay, so these are both examples of gene mutations, and then the other type of mutation that I wanted to talk about today are chromosomal mutations. Okay, chromosomal mutations. And so instead of being a mutation that affects just one gene, it's a mutation that can affect a whole chromosome. Okay, so for this we have to think back to meiosis, right? Meiosis, again, was the process that organisms use to produce sex cells. Okay, and during a stage of meiosis, during meiosis 1, we have a part where the chromosomes that you have line up in what's called homologous pairs. So like the same chromosome, but the ones you get from both parents line up. So let's say you have a chromosome from dad, right, and we'll use blue for that color, and then it has replicated, so it's in that kind of X shape, right, and it's connected together, and it lines up next to the same chromosome, but the one that you got from your mom. Okay, so now we have these two chromosome pairs lined up, and so one type of mutation that can happen is a translocation. Okay, and really what that means, or let me rewrite that. Translocation. And what that means is that part of one parent's chromosome can just move to a different part without trading back a little piece of the chromosome. So what can happen is, let's say this little piece of dad's, it just falls off of his chromosome and attaches to mom's. Okay, so what can happen is, you have this one chromosome here, and then Now it's just missing that whole piece. And so mom's chromosome, she has all of her DNA on it, and then she has that little piece of dad's extra on here as well. Okay, and so this is bad because now this chromosome is missing a lot of the genes that were stored right here, right? And this chromosome has some extra genes here. And so if these chromosomes, which are in your cell, are used during meiosis, this faulty chromosome can be passed on to the sperm cell or the sperm or egg cell that you're making, right? And so you can pass on this one in a gamete or sperm or egg, and it's missing a lot of the critical genes here. And so you'll have some kind of mutation. You might have some mutations in the offspring that's made where they're missing certain traits or certain genes or certain proteins. And then same thing for this one. This will have some extra here because it has the extra from dad's. And so that'll also cause some complications in the offspring that's formed as well. And so this is a translocation. It's just where a little piece of the DNA moves from one chromosome to the next. And then the other chromosome mutation that can occur, it's what's called non-disjunction. Okay, so non-disjunction. And if you look at this word non-disjunction, disjunction just means, separating. And so it happens when chromosomes don't separate. Okay, so let's say we start off with those homologous pairs again, right? We have, dad's chromosome here, and then we have mom's chromosome here. Okay, and so normally what would happen is, these two chromosomes would separate out, and then you'd make eventually, 4 different gametes, right? So like 4 different sperm or 4 different eggs, during the whole process of meiosis, and each one would get one single chromosome of these 2 pairs. So like we could get 2 blue ones here, and then 2 red ones here when they separate out. Okay, but non-disjunction, they don't separate. So now, what can happen is, this one will, or they don't separate properly. So this one will be here, and maybe when it does separate, and let me just draw it out first. Here's mom's chromosome, and this is a pretty bad drawing, but you get the idea. Okay, let's say that, instead of separating out properly, these 2 chromosomes here don't separate, or these 2 chromosomes here don't separate. So what can happen is we'll still make 4 cells, right? But the cells will have the wrong number of chromosomes, right? If we can think about these 2 blue ones not separating, then now this chromosome will have, or this gamete that's produced will have 2 chromosomes instead of 1. This one won't have any of the blue chromosome here, or any chromosome at all, and then let's say this red one separates fine, so it's normal. And so what can happen is if this gamete with an extra number of chromosomes fertilizes with another gamete with the same number of chromosomes, then the zygote that's formed, the zygote being the fertilized egg, will have the wrong number of chromosomes, right? And that can cause some complications. It can cause a miscarriage or some of the genetic disorders that we've talked about earlier. So these chromosomal mutations can also have changes in the traits and the proteins that are passed on to the offspring as well. And so just to summarize what we talked about, remember we're talking about mutations, and mutations is just a change in an organism's DNA. And the 2 ways that really they can happen is if it happens in a single gene, a gene mutation, whether it's by a substitution or an insertion or a deletion, or it can be whole chromosomes, right? And so we can have a translocation where part of the chromosome moves to another chromosome, or we can have a non-disjunction event where chromosomes fail to separate properly. And these will have different kinds of effects based on what's happening. But basically, the idea that you guys should have is that the DNA is what's controlling your traits, and so changes in the DNA, whether they happen in gene mutations or chromosomal mutations, will affect what traits that organism is going to have. And these mutations, they will only be passed down if they happen in a sex cell. So if they happen in your sperm or your eggs, they will be passed on to the next generation. If it just happens in a normal body cell, it might get passed down to other body cells in your body, but it won't get passed down to your kids. Okay, so these mutations only get passed down to kids if they happen within your gametes, so your sperm or egg or your sex cells.